Thank you. So today we'll be discussing on drug design. I'll be doing some demonstrations as well, some hands-on demonstration, and I'll be taking us through the process of downloading the softwares that we'll be needing for the training. Before we start, I would love us to have a word of prayer before commencing our today's training. Father, we thank you for giving us the strength, the ability for us to gather once again to learn. Father, even as we learn, we pray that this session be an impactful one and we'll learn a lot, the things that we needed for our personal development and growth pertaining to this topic. We pray you teach us and help us to understand every little thing of it. In Jesus' name we've prayed. Amen. Thank you once again. So, as we all know, this is GeneCraft Microbial Genomics Academy. The academy was founded February this year, 2021. By next year, February, we'll be celebrating our 1-year anniversary. And the sole purpose of founding the academy is to give access to individuals that would love to know, to learn, to unlearn and relearn in the world of bioinformatics. And our first cohort, we started by, I think, May. We started and ended around June, July, August. So, and most of our participants gained the necessary skills and knowledge they need pertaining to this. And by God's grace, they've also done some manuscript publication work from it. And presently they are undergoing reviews in respective journals. So we are grateful to God for the strength. So our second cohort, we decided to start with molecular docking. And subsequently by January, there will be another training section that will be totally on bioinformatics for disease diagnosis. This one is bioinformatics for drug design. In January cohort will be working on bioinformatic tools and databases that we can use to predict genes or to predict to understand the functional genomics of certain biological database. So welcome to our second training, our second class, where we'll be taking drug design. Then for other trainings that we are currently undergoing, that is to start, that will be on Sunday, this week Sunday. The onboarding session is this week Sunday, geospatial mapping in public health. This training is aimed to like give the knowledge, the necessary knowledge that people need concerning how they are to analyze your spatial data, especially in mapping disease areas and maybe to search for hotspot areas for particular disease trends. So make use of GIS tools in order to map these disease areas. If you are interested in joining this cohort as well, you can private chat me on WhatsApp. So let's go to our the main for today. Hope you can hear me. Let me clarify myself. Hope you can hear me.
Yes, ma'am.
Okay. So we all know drugs, and without drugs, majority of the illnesses that are occurring, we give a detrimental effect to individuals. There will be high death rates and there will be no solution to some medical problems. And then this targeting means of trying to kill or inhibit or downregulate certain enzymes or certain regulatory bodies that are involved in disease. Paul Ehrlich, a man named Paul Ehrlich, he was actually the first man that brought about targeting of diseases using a particular molecule or using a particular substance to target a particular disease in order to either stop the function of that particular disease or to inhibit the activities that are involved in that particular disease. So a drug is any molecule, any chemical substances that when administered, we actually change or produce some biological effects. And these biological effects can be good ones, but when it's being misused, can be on the other bad side. So Paul Ehrlich brought about this target system, and he said he brought about the system in which that if there can be a particular molecule that could target a protein, I think he called it, was it a bullet point method. That means if can be a particular molecule that can target a protein that is involved in a disease or that is involved in other regulatory pathways, that means we'll find cure and we'll also find enhancement to some of them. Now, you will agree with me that there are many proteins in our body, and individually, these proteins, they perform specific functions that will help in the everyday activities of the body. Now, there are some proteins, when they are not well secreted, there is a problem. When they are over secreted, there is a problem. That's why balancing has to occur in any of our body system. There must be a balancing mechanism so that we'll not have overproduction or underproduction. And peradventure, if we should have an underproduction, there should be a target that whenever that target should meet up with that particular protein, there can also be some sort of activation. This is when we come with respect to maybe with respect to diabetes, maybe when the cells or the cells in the pancreas are some certain cells that are found in the pancreas are not producing or are not producing insulin or are not metabolizing sugar very well. You tend to see that this sugar will be overproduced in our system because there is no means by which those sugar can be broken down. Those things that happen to some people might be as a result of the inability of some certain cells to produce the enzymes that are needed for the activation. So these bullet points can come in the sense that, okay, why not I produce a system by which whenever it targets or meets up with this particular cell in the body, it can help boost its efficiency. It can help boost its method of action so that by the time an individual should take it in, it helps in boosting it, and that boosting will now help in making that cell to work effectively. And these are actually with the help of some biological pathways and biological regulatory systems in our body. So that will now bring us to drug design, a method by which we create new or novel drugs that will target specific enzymes or specific molecules in our body that can now like promote good health in every system. We develop new drugs every day in order for there to be advancement or enhancement in the health sector, in order for diseases to be well cured, in order for other mechanisms by which we can use in supporting some diseases that can't be cured at all. So drug design is necessary because everything God made, He made it good and beautiful. And in everything that He has made, there are also some bioactive components that can aid. Most of these bioactive components, some of them have not been found up to now. That's why research is there. Research is ongoing to sort for new bioactive compounds, because it's more like the ones we have, although they are working, but due to resistance rates, due to some tolerance from the organisms, they tend not to work effectively as when they were originally produced. So there is a need to discover new drugs that can support the other or replace those ones as well. So that's majorly was drug design. But the thing is, majority of us don't really understand protein. When I say protein, I don't know if we have a clear perspective or a clear understanding of what a protein actually is. Can someone tell us what a protein is? Just an idea, if you have any. Is anyone on the call?
Hello? Yes. Protein
Okay.
is the combination of amino acids joined
Okay.
together to give a
Okay.
functional compound or a functional molecule called protein.
Okay. Thank you very much. You said it all. And as he said, proteins are building blocks of amino acids. And how do we get amino acids? We look down to the blueprint of life, which is the DNA. If you see my slide on the left-hand side, you will see a molecule, this one, the X chromosome, the chromosome, which symbolizes the DNA of every individual. Now, inside this DNA are genes, are sequences of nucleotides that coupled up to code for a particular features, or these features can either be physically seen or they are inherent, they are inside an individual, but some of them are not expressed. If we go back to our biology days. Now, in every individual, we know we have 23 pairs of chromosomes. And these 23 pairs, they are present in every individual. Down part of this chromosome, you see the different 23 pairs of chromosomes that we have. Now, when some of them are like over-secreted, there is a problem. For example, we have this chromosome 21. This chromosome 21, they perform some biological functions in the body. Now, you can have a pair of chromosome 21, but there are some individuals that this chromosome is overexpressed. In other words, instead of you to have a pair of chromosome 21, we are now having three different not pair this time around, but three different chromosomes of that particular 21. That extra chromosome that is produced in that 21 can lead to what is known as the Down syndrome. You get? So that's where balance, that's why I talked on with respect to balance. There must be balancing in our system. So when they are produced, they should not be over. When they are underproduced, there is a problem. Now, that Down syndrome can lead, is a particular disease that is a genetic disease. I think scientists have even found a way to like find means of deleting that extra chromosome that individual or that might be present in the DNA of that individual. Now, but before we get proteins, we look at the blueprint of life, which is the DNA. The DNA is the basic unit. And inside this DNA, for a protein to be produced, we have an RNA molecule. From this, my right template, you see what is known as transcription, and transcription takes place when an RNA molecule copies the genetic information from a DNA. And there are actually three processes, I think four processes. We have initialization, elongation, and the termination stage. All this will be explained in our next cohort. All these are majorly concerning bioinformatics for disease diagnosis, so we don't really delve deeper. But the information that is being copied from this DNA, they are transcribed, they call it transcription process, into an RNA molecule. This RNA molecule now translates, it's more like interpreting what is being copied from the DNA molecule. And whatever is being translated are now what is called what? Proteins. Those sequence of amino acids that they are being translated in after some several steps. Even in this translation step, we have two major steps. After that, the proteins, the amino acid sequences are now folded into what is known as the protein. So the protein are now the building blocks of this system that we have today. Now we have proteins that can be found in organisms, and these proteins that are being produced are for pathogenic reasons. We have some proteins in our body. When they are overproduced, there is a problem. So when you see proteins, don't just see them as, although they are bad, not all proteins are bad. But the ones that are bad are the ones that we now target. We now find means or ways by using bioactive compounds to target them, to see if there can be some exchange or coupling, just like I explained in our first class, if there can be a kind of a binding affinity between the bioactive compounds and the proteins that are being targeted, or that the proteins of interest that we feel or have been researched to be concerned with pathogenic activities. So that now brings us to what is known as the processes in drug design. We majorly have two processes. We have the structure or ligand-based drug design, just as I've explained earlier last class, where we can predict the binding affinity between a known ligand, unknown protein. We don't know the function of this protein. We just want to check if this known ligand can bind with this particular protein that we don't know. Now, target-based drug design is actually a process of discovering new drugs by searching and understanding the proteins itself. That's target-based. We know the function or the enzymes that are being produced by this protein, and we want to check for the druggability, the binding affinity of this particular protein with a known ligand as well. That's target-based. Now, we now move to the basic computer-aided methods that we can use in drug design. We have target identification and preparation. That is where our next class, after downloading these softwares, we'll be doing protein and ligand cleanup. We do the preparation by cleaning them up, removing some extra molecules that might be present when we download the 3D structures. By the time we download the 3D structures, there are still some molecules or ligands that might be attached to the structure. So the first thing we do is to prepare them by cleaning them up, so that we know that whatever we are working with is the protein direct or is the ligand direct. Then we select our ligands. It can be any bioactive component or a drug of interest against a targeted protein that you know of. I think there is a particular plant called dogoyaro. I don't know if you know of neem plants. That plant is known to cure tuberculosis. I think from traditional knowledge, is known to cure tuberculosis. And some of my students, they worked on the bioactive compounds from that particular plant, and against the protein for tuberculosis, there was a good binding affinity between them, and the docking process was really a successful one. That was their group project. And we have different databases that we can use in downloading these 3D structures. And we know databases are like biological software systems by which a lot of data, biological data are stored. So by the time you go to this site or website or the database, you'll be able to retrieve, because there's nothing new under the sun. A lot of researches have been going on, and a lot of data have been deposited in these biological databases. So by the time you search, you'll be able to like retrieve some for use with your system. And we have different types. These are we have primary database, secondary database, and derived database. The primary database are majorly for searching for genome sequence in the sense that we have, I don't know if you know of genome sequence, the nucleotide sequence of an organism, or the DNA sequence of an organism. Then if we want to like talk about the structures, we come to secondary databases and some other metabolites. Maybe metabolites that are produced during the process. What is this protein involved in? What are the mechanisms? Can they interact with other protein? Do they cooperate with other protein to actually cause the intensity of this disease? We now looked at derived databases where we can get the biological processes, databases where we can study the biological processes of some of these proteins. Now that brings us to, for database for protein, for ligands, we have the PubChem. These are the sites. We have ChemDB, we have Zinc, we also have Drug Bank, of which we'll be exploring as our hands-on. So for the protein modeling, this has to do with predicting the three-dimensional shape of protein molecules. Maybe, for instance, if you should do a sequence analysis, maybe from your lab, after isolating the organism, you did the molecular identification, and you got a protein sequence. How do you predict the structure? How do you know the structure? How will you be able to retrieve it from an online database? We are talking of protein modeling. When you are trying to predict the 3D structure of proteins, it's protein modeling. And we have majorly two types. We have the homology modeling and the ab initio modeling. The homology modeling is more like the proteins have been, you are trying to like check the similarity of your protein with existing ones. So if maybe you should have a protein sequence that have been worked on before, you can easily get the 3D structure by working with a similar template. For example, this tuberculosis gene, there's a particular gene called DPROE1. That DPROE1 has been like worked on and have been deposited on protein database or protein structure databases. So by the time you should do some isolation and some sequencing and you find that particular gene, you'll be able to get the 3D structure by searching for that particular gene on the data bank, or there is a particular web tool they use, AlphaFold. So that AlphaFold can help you to predict the 3D structure, because others have worked on it and it has been predicted by this artificial intelligent data setups. So you just go, type in the name, and it will bring out the 3D structure for you. But there are sometimes you have a protein that has not been worked on. There is a particular, what's the name of this data bank? that is sponsored by AlphaFold. Yes, we have the Google Deep Learning, Google DeepMind. They were the one that actually brought about AlphaFold. I don't know if you've heard of AlphaFold before. How many of us have heard of AlphaFold? Can someone say something? Hope you are listening, or I'm just talking to myself.
No, we are listening. We are listening.
Okay, there is a kind of a group. They call them AlphaFold, and this AlphaFold was originated by a Google deep learning model. I think they published their work on Nature. So they came about and said there is a means by which we can actually predict proteins efficiently using a computer and their web tool. So although initially, before we can predict protein 3D structure, we have to do some other laboratory analysis that has to do with I think radioactivity um, that has to do with radioactivity works. So before you can predict a protein structure, you do that in the lab using machineries. But now these individuals came up as a team and built up a system by which we can easily predict their structures on a system online without you doing other laboratory analysis. They published their work on Nature. I think that was it 23rd of July 2015 or so, abi 15th of July. That was when AlphaFold, the name started sounding. Now I have a protein sequence that have not been worked on. They've not predicted the 3D structure. How can I go about it? That is what is known as the ab initio modeling. The protein structure that have not been predicted. So you can actually design a protein fold using another web tool and database. That one is called AlphaCollab. It's another database that was also produced by these individuals. So first off, I want us to go to our hands-on practical where we'll be downloading these softwares. Hope you are with me.
Yes, ma.
Okay, for the Chimera X, we'll be using three softwares. We'll be using Chimera X, we'll be using Pyrex and Discovery Studio. For the Chimera X, this video will be uploaded. So by the time you watch, you'll be able to download it yourself. You just search, go to Google, Chimera X, download. This is the one you will pick. I've downloaded this software on my system, so I don't want to re-download it again. Please, let's be patient. Why is it taking long? Any questions so far as we are going through the software download? Any questions? Lovely. Any questions so far? Success, did you understand what I was explaining?
Yes, ma. Yes, ma. I'm following.
What of checking out? Have you been following what I was saying? Good.
Yes, ma.
Is there any question from your end?
No, ma.
Okay. Abu, are you with us?
Very sure. Yes, I'm here.
Okay. Please, I don't know what's wrong with my network. Can you imagine? I'm coming. I'm trying to see means by which we can download them. Just go to Google Chimera X. Download. Okay. So you click on this first one. Please, let's hold on. My network is glitching. Let me see if it's the site. I'm coming. Let me search for Pyrex. Okay. For the Pyrex, this is the one we'll be downloading as well. But Pyrex normally takes time. So when you are downloading your Pyrex, ensure that you have enough data and time. Is it loading? You go to, for the Pyrex, after you've loaded it, it will give you this interface. Then you come to free download. You choose, you understand? You click on the choose for free. Then it will take you to another section where it will be showing you that it's already downloading. So that's for Pyrex. Then let me see for the Chimera X if it's coming up. Then for Discovery Studio, you can click here and search for Discovery Studio. It will take you to this. Okay, I did not put Discovery Studio download. Discovery Studio download. Don't know why my network is bad. So it's a BioVar, it's called BioVar Discovery Studio. So by the time you click on it, it will show you a place where you will need to sign in with your Gmail for the Discovery Studio before you go ahead and download it. Share my I'm coming. Hope you can see my screen.
Yes.
Okay. Chimera X. Download. These are things we can do ourselves just for you to know. I will type the name, so you just go ahead and download it yourself. Then the network is coming on and off. For the sake of our time, if we navigate through the site, we'll see ways of downloading them. I don't know why it's now my network wants to click. For the sake of our time. Now let's go to means by which we can search for proteins. How do we search for proteins? We have protein data banks, and we also have, I don't know if you've heard of NCBI. You can search for your protein molecules from NCBI as well. For ligands, we do PubChem. You can also search PDB as well for protein data, for proteins. For NCBI, let me show us the method we can use to do that. For NCBI, go to NCBI, you click on it, and you click on this first result. It will lead to their database where you can search for proteins. There are different tools that you can use, and there are different data sets that have been documented in their site as well. Please confirm if you can hear me.
Yes, ma'am, we can still hear you.
I'm coming. I want to change my network. Let's see. Okay, let's check UniProt, PDB, Protein Data Bank, PDB. Okay, this home page for PDB. I have a particular protein in mind that I want to search for, the DPROE1 protein. That is so bad. I've been using this thing since now. Okay, this is the PDB interface. Protein PDB stands for Protein Data Bank, where you can retrieve your proteins, protein sequences, and if you have a sequence you don't know the name, you can also blast it on protein database PDB. Jesus Christ. Please give me some time. Give me some time to rectify this network issue. Let me change to another server. Please Let's take
Okay.
5 minutes break. I'm coming back ASAP.
Okay, ma.
Let me change my. Can't I? Hope you can hear me now. Please, I'm so sorry. Can you all hear me now?
Yes, clearly. This camera can be downloaded today because it's very heavy, almost 360 something MB. Hello?
Hello, can you hear me?
Yes, ma'am, we can hear you.
Okay, thank God. Okay. Today, okay, so let's go back to our Chrome. Let's see. Okay, this is the NCBI. Let me do it again. You search for NCBI. This is the database where you can retrieve biological data set like your DNA, protein sequence, some other RNA sequence as well. So when you come to this interface, you come to all databases and you click on protein because what we are searching for at this particular time is a protein. So you click on protein, then you type the name. I want to use DPROE. It's a gene in tuberculosis. DPROE1 protein. Okay, so it will show me varieties of DPROE protein. So from our searches, you could see that they are all protein affiliated with Mycobacterium tuberculosis. And you can decide to pick any of them. I think I picked the first one. This is showing the amino acid sequence, the number of amino acids that are present. We have 466 amino acid sequence. And for us to download this, we'll click on it. You can decide to pick as many as you want, but we want the FASTA file. This is the FASTQ file. FASTQ file is a kind of a file extension for NCBI where the information about the sequence, everything about the sequence, the people that carried out the laboratory analysis, the organism where they were sequenced from, the samples, the topic, the title, the authors, and everything are contained in the FASTQ file. But in FASTA file, or they call it FASTA file, the FASTA file is only concerned with the either nucleotide sequence or the protein sequence. You can see that there will be a difference between this. This is the FASTQ file. Now let's click on the FASTA file. You see that the FASTA file is only showing you the nucleotide sequence. And it's usually a FASTA file is usually recognized by this greater than sign. Once your FASTA file is not having this greater than sign, you will not be able to like do some analysis in bioinformatics. But for maybe searching for proteins and the rest, you can be able to do that. So what you need to do is maybe we want to check for, we want to predict the 3D structure of this protein. You can just decide to copy only the protein sequence. Hope you can hear me. Can decide to copy only the protein sequence. Hope you can hear me, please.
Yes.
Yes, ma'am, we can hear you.
So after copying, I told you that we can search for proteins with NCBI database. Now let's go to another database, PDB, Protein Data Bank. PDB. Now this is Protein Data Bank. You can also search for nucleotides here. Okay, here you come. You enter your query. Your query means the activity you want the database to do for you. So I can decide to enter, maybe as I have that sequence, it's either I enter the sequence or the name. Let's see if we'll see it here. DPROE1. DPRO stands for dioxygenase. I think we'll see the name now when we search for it on PDB. Okay. In this interface, this section is showing everything about the protein sequence. You know, I was telling you that you can actually predict the structures with radioactive laboratory analysis. You can see that the experimental method that they use in getting the fold of this protein is X-ray. So one of the protein that was deposited was actually predicted using X-ray diffraction method. Now, this shows you everything about the protein, the left-hand side, the scientific name, the bacteria, where it's gotten from, everything you need to know about the protein is here on the left-hand section. Then we also have a code for each protein that have been deposited. We call them the PDB code. So this is the PDB ID for this particular protein. But the name of the protein is, okay, this is a kind of the structural name of the protein. But it's called, okay, yeah, macromolecule decafrenyl phosphoride beta D ribose oxidase. So that's the name of the PDB, the DPROE. That's the full meaning. And the method they used was X-ray diffraction method. So you can see that I told you that initially that when you download proteins, we have to prepare them by removing some ligands. You can see now that there are some ligands that are attached to that particular protein. So by the time we download it, we have to remove these ligands and some other water molecules, because we will not want the ligands that have been synthesized with it to work with our own ligand. We want to test our ligand directly with the protein. So we have to remove these ligands by the time we download them. So you can actually download them in PDB formats. Now, how do we download file? Okay, let's see. Let's click on this. So you can come here and download file. You search for download file. You will want to download it in a PDB format. So you can click on here and it will come to it will bring up another icon where you can now press save. So it will download. See, so these are this is for PDB. I've worked on NCBI. Now let's see how we can download ligands. Ligands are our drugs or bioactive compounds. We also have data banks where all these things have been stored. You can do that in PubChem. So you come to PubChem. By the time you search for PubChem, let's what ligand can I work with now? Oh, I don't know a particular. Okay, let's work with aspirin. Aspirin is a drug. So let's work with aspirin. You search for aspirin. You see the structures about aspirin. You will see a lot of results that have been worked on aspirin. a lot. So it depends on the one you want to work with. We can decide to pick the first one. So by the time you check, you see the 3D structures of this drug as well. So here is the 2D structure. If you click on the 3D structure, you'll be able to view it very well. Okay. You can also change the dimension, either ball and stick, or, sorry, hope you can hear me.
Yes, ma'am, we can hear you. We're following.
Okay. So those are the two major methods we can use. PDB for protein or NCBI for protein. Then do PubChem, use PubChem for your ligand. That's that. Now we already have our, I want to speed it up because the power is going off soon. For instance, since we already have our protein sequence, we want to like search for it in AlphaFold to see if it has been worked on before. You just search for AlphaFold. AlphaFold. To take you to AlphaFold is where you can search, where you can do your homology modeling. In other words, you want to use another person's template to like get your own 3D structure. So we search for the DPRE1 protein. Now it will show you the structure. It will show you different results, and you can pick the one of your desire. So here we'll see the fold, the 3D structure very soon. Okay, so here's the 3D structure of the protein molecule. How do we check for this now? I'm coming. Okay. So you can also download your 3D structure here to get a better view. Okay, to get a better view, you increase. These are the tools you can use. You can decide to increase or reduce it. Hold on, coming. Okay. You can use your cursor to be increasing or reducing to check for the dimensional and the different sides of the protein sequence. Now our assignment will be, because since we'll be working with camera X directly, I would want us to search for ligands that we can use, ligands of interest that we feel we want to work on, and protein of interest we feel we want to work on as well. So that by the time we are downloading them, we'll have to still do some downloading next week. I have my ligand of interest, I have my protein of interest. So you can go home, search for them, download them, and have them on your system. So that by the time we start our practical session using Camera X, you'll be able to understand, know how to import these structures into the software that we'll be using. Now I have, for instance, I have a protein sequence that have not yet been known. The 3D structure have not yet been predicted. How do I do that? You can come to AlphaFold Collab. This is a work I've done. This AlphaFold Collab will help you to predict the 3D structure of the protein. So this work have already been predicted for me. Although this protein is known, I just tried it to see if it will help me predict it, and it gave me some results. AlphaFold Collab is a database or a software where you can predict protein structures that have not yet been known, or protein structures that have not yet been predicted using your own personal sequence. You use AlphaFold Colab. In our next class, because it takes time too, I will search for a sequence that maybe it's not common, and I will try it. I will run it on the AlphaFold Colab, so you'll be able to get a very good understanding on it. Any question at this particular time? Please, room for
So,
questions now.
excuse, so that definitely now what you are saying is this alpha fold is for homology modeling. So
Yes,
that
for
it
homology
means
modeling.
if we have a protein sequence, so yeah, it is a molecular docking, but we have to start generating our own this thing, our own
Code,
3D
3D dimension.
yes.
Yeah.
So now you teach us the homology, or you just showed us that in case you have a
Yes,
separate class
I will
for
teach
homology,
you.
just like now that we have a molecular dynamics is also a separate class. So this is what I
Yeah.
want to get to understand.
Yeah. This particular AlphaFold collab is for AB. I used to call it AB initio. I don't know if I'm correct, but others call it AB initio. So, like, I have a sequence that I don't know the fold, or maybe the fold or the 3D structure have not yet been predicted. You understand?
Yes.
In our next class,
Mm.
I'll be, because the process is really long. You can see that I think this is what even drained a lot of my data. It took me an hour to get to this result. You
Okay.
understand? So, and the sequence I used was a known sequence. It was still that DPROE1. But
Okay.
let's if the power will go with that. Now, you search for alpha fold
Mm.
collab. Okay. Now, this AlphaFold
Mm.
collab, you click on it, this particular one. For you to install AlphaFold, I don't even have it on my system. It takes a lot of memory. I think you
Okay.
should have enough memory to get the software. But the good thing is that
So
you can use
online.
it on Google. Yeah. And this is the
Okay.
Google link. If you want to install it, you cannot install it from this site as well. It's loading, thank God. Now, the first thing you do here is to, I don't know if you are seeing my cursor. Can you see my cursor?
No, the cursor just went out of.
Can you see it now?
No, no, up to now.
Yes, I can see it.
Okay.
Okay, you
So
can
you
see
come
it.
to this place. There's a place called Connect. Can you see Connect?
Yes.
So you have to connect it to your RAM
Okay.
so that it will work in accordance to your computer storage. So it's showing connected. You can see that it has connected my RAM and the disk.
Okay.
Hmm.
Okay.
So this is where you now put your query sequence. You remove this your query sequence
Mm.
and put in yours. I think I still have that my query sequence, that DPROE1 sequence copied. So you delete that query sequence and put yours. And remember, that one that is yours is the one that you don't know the 3D have not been predicted. But how will you know if the 3D have not been predicted? It's when you search on that PDB. If you search on PDB, you did not see it's not yet
Mm.
found. Just know that it has not
Mm.
been, the 3D structure have not been
Deposited.
predicted. So you come to
Okay.
runtime. Can you see my cursor?
Yes.
You come to runtime. Then you have to change the run type. I don't know, but sometimes I think this one it came with default setting. So you can change your runtime, and that runtime should be T4 GPU. Are you following?
Okay.
So you save. Are you guys following me?
Yes.
Okay. Then after doing that setup, you can come to run all. It will give you this notification. Just say run anyway. So it will be
Okay.
running it for you. It will take time. Now, I don't want it to do that same process for me. I can now come to my
Okay.
results.
Okay.
I will leave the site so that I'll come to my results now. I think this took an hour for me. So it has
Okay.
folded. These are the lapses, the running percentage by which it finished it. So if you go down, if you scroll down, it will be displaying the running percent, the progress. You understand? It will be displaying the progress of the running. Now, this is the sequence
Okay.
coverage. And here is my fold. Here is my fold. Yeah, here.
This is ready now.
So it has folded
Okay.
the protein sequence for me. I think there is a place you can download this particular fold. Still navigating the sites. Coming. Here is the place again. It's also showing us the quality score.
Okay.
The blue
Of
stands
the.
for yes, of the prediction. That is the accuracy.
Okay.
So you can see that we have much blue to tell you that it's really it's kind of not even exactly the fold, but it's accurate to some extent.
Okay.
Here are some plots and graphs that you can use for your publication as well. You understand? These things, you can find them in publication based on sequences that have not yet been analyzed or deposited. So maybe if you get a result like this now, you cannot deposit it on the AlphaFold
On 3D.
database
Okay.
so that anybody that will work on an identical sample like yours or get an identical sequence like yours will now take your own that you submitted as the result will now come out. Your own will now come out as a result of what he has submitted. In other words, his was similar to yours because you deposited it on their site.
Okay.
So any further question? At this time, I'm entertaining questions. Please, please ask questions now. Is the class, are we good? Are we good to go? So my questions for Aminu, what have you learned so far from the class? Aminu, are you with us? Success, are you with us?
Yes, ma. Yes, ma. I'm here.
What have you learned so far? Hello? Can someone summarize what we've learned so far today?
Okay. From what I was able to gather today, we talked about drug, what a drug is, basically a substance that is administered to produce a biological effect. We talked about the processes of drug design with the ligand-based drug design and the target-based drug design. Then we started talking about the databases the ones for the ligand, the ones for the protein, and yes, and then the databases that we can use to for the protein 3D structure modeling. Yes,
Okay.
that's what I want to gather.
So what do you understand by homology modeling?
Okay, homology modeling is like is a type of a protein 3D modeling where the 3D structure is predicted based on similarity to other existing protein sequences
Yeah.
or other existing structures. Yes.
Okay. What of ab initio?
Ma?
What of ab initio?
Okay, this is the type of modeling where the 3D structure is predicted without a known existing structure that is similar to it.
Existing structure. Yes, yes. Okay. Any other question before I go? Please, let's hold on. I'm trying to take some pictures for the class. Please, some of us are not following us on LinkedIn. Please, let's do the needful, please. How many of us are not following us on LinkedIn here? The class will be over soon. Just some pictures. Then in our next class, I even wanted to ask, I don't know which day would be convenient. It's as if that Tuesday, Thursday is not really convenient for some of us. I don't know. Am I speaking the truth or not? If it's not convenient, let me know how to shuffle it again. Are we still in the class?
Yes, we're in the class.
Is the days, are they okay? Or we should
Yes, my
change it
dear.
to the days are okay, right? Because I wonder
Yes.
why many people don't come to class, and we are up to, I think, 30 something in the group.
No, the only thing I will suggest is maybe you do a poll and set it in the group. So
Okay.
for some of us to answer it, yes. So the highest number
Or
of
no.
days that are selected, then you will just, if you see as the teacher it's okay with you, then you go with it. Me, I feel this
Okay.
will help with
Okay. Then for our payments, we are not really responding to it. And our practical section will be starting officially on Thursday, which is
the,
tomorrow.
yeah, since
But
we have class yeah, since we have class tomorrow, but from tomorrow, maybe we have Friday, Saturday, Sunday, Monday. Within the 4 days, at least you'll be able to have seen the result of your poll and decided by Monday if it's going to continue on that
okay.
Tuesday or Thursday or any other day that you feel is okay based on the poll. Then we are good to go, I guess.
Okay. No problem. I'm almost done. Please. I'm almost done, please. Just one more last shot. Ah, thank God, they've even brought lights. Then from next week, we'll be having our facilitator, a new facilitator. He'll be the one taking us the in-depth aspect of the course. So let's not miss our classes next week at all. We'll be doing Kirk pathway analysis, I think network pharmacology, to know the metabolic measures by which this protein actually carry out their responses and the rest. Okay. One more last. I think attendance is compulsory as well. Okay. Please. Okay. Thank you for attending. God bless you and have a nice night's rest.
Thank you very
Thank you
much,
very
ma'am.
much.
Thank you very much. You're welcome. I think I cannot stop.